Hello everyone, this is my video of introducing semi-conservative replication of DNA to you. It is a theory developed to describe what happens when the DNA is being replicated. Let's find out. Replication of DNA is used in cell division because newborn cell needs an identical replicated version of DNA from his parents. The concept of semi-conservative replication of DNA is very straightforward. We all know that the DNA has a structure of double helix made of two polynucleotides pairing with each other. So semi in terms just means half, which is one polynucleotide, and conservative means to be kept same. So the idea is to firstly separate two strands of DNA, which is stated by the red column in my model, and then pairing them with two additional polynucleotides. In my model it is yellow. So after semi-conservative replication of DNA, the two products are identical and half of each of them is from the original DNA and the other half is added during the Here is a detailed explanation of how the process is like. The DNA firstly unwinds itself and the hydrogen bond breaks. Then there are nucleotides that is free moving in the nucleus, and some of them with two extra phosphate are activated. At my model, I present them as two sponges. Next, the DNA polymerase enters the game. It links the half chain with a new nucleotide around that is complementary to the original polynucleotide. As the DNA polymerase moves forward, more and more base pairs are produced. Because there are multiple primer and DNA polymerases works in one completed replication of DNA. So when the DNA polymerase's work is done, the enzyme exonuclease removes all the primers from both new strands of DNA, and another DNA polymerase then fill the gaps, and lastly the DNA ligase seals the whole strain and the replication is done. In 1958, Matthew Matheson and Franklin Stahi carried out an experiment to verify the hypothesis of semi-conservative replication. Through incubating E. coli in food containing different isotopes of nitrogen, the composition of DNA will be very different after the replication. Due to the fact that bacteria grown with nitrogen 14 is lighter than nitrogen 15, they discovered that the set with only one replication in nitrogen 14 food has the same amount of nitrogen 14 contained DNA and nitrogen 15 contained DNA. And as there are more replication took place, the amount of nitrogen 15 DNA decreases as the nitrogen 14 DNA increases exponentially. They realize that the DNA replication is done semi-conservatively. DNA polymerase itself has a mechanism of proofreading. It only pairs the nucleotides that is complementary to each other and has a function of excising mismatches. In this way, mutations have very little possibility to happen. Hope you guys enjoyed this video and see you later.